Ncert Reading, Class 12, Chapter 2, Sexual Reproduction in Flowering Plant. Are we not lucky that plants reproduce sexually? The merits of flowers that we enjoy gazing at, the scents and the perfume that we swoon over, the rich color that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structure of the inflorescence, flowers and the floral parts shows an amazing range of adaptation to ensure formation of the end product of sexual reproduction, the fruits and the seeds. In this chapter, let us understand the morphology, structure and processes of sexual reproduction in flowering plants. Angiosperm. Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are object of aesthetic, ornamental, social, religious and cultural value. They have always been used as symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In class 11, you have read the various parts of a flower. Figure 2.1 will help you recall the parts of a typical flower. Can you name the two parts in a flower in which the two most important unit of sexual reproduction develop? Pre-fertilization, structure and events. Much before the actual flower is seen on a plant, the decision that the plant is going to be flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a wall of stamen representing the male reproductive organ and the gynoecium represent the female reproductive organ. Stamen, microsporangium and the pollen grain. Figure 2.2a shows the two parts of a typical stamen, the long and slender stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamen are variable in flower of different species. If you were to recollect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is their dithecus. Figure 2.2b. Often a longitudinal groove run lengthwise separating the theca. Let us understand the various type of tissues and their organization in the transverse section of an anther. Figure 2.3a. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided tetragonal structure consisting of 4 microsporangia located at the corner, 2 in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers. Figure 2.3b. The epidermis, endothecium, middle layer and tapetum. The outer 3 wall layer perform the function of protection and help in the dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. It's microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrad. What would be the ploidy of the cells of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspore from a pollen mother cell through meiosis is called microsporogenesis. The microsporogenesis as they are formed are arranged in a cluster of 4 cells, the microspore tetrad. Figure 2.3a. As the anther mature and dehydrate, the microspores dissociate from each other and develop into a pollen grains. Figure 2.3b. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. Figure 2.3c. Pollen grains. The pollen grain represent the male gametophyte. If you touch the open anther of hibiscus or any other flower, you would find the deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, size, shapes, colors, designs